Okay, trying. Okay.
Perfect.
Okay. Well, I was first of all relieved to see everybody here today because it wasn't clear that all of you would survive last week, but it looks like you did. Okay, so Friday you learned how to do atmospheric correction. Take the top of the atmosphere radiance out of your satellite. You finally get it down to a water leaving radiance, or generally its equivalent, some kind of water leaving reflectance. So now this week we'll talk about how do you take that reflectance spectrum at the sea surface after you've atmospherically corrected and got rid of all the bad stuff like aerosols and absorbing gases and sun glint and all of that. How do you take that spectrum and now extract something that you're interested in, like the chlorophyll or the absorption at 400 or the bottom depth or whatever it happens to be? So I'm going to give you a lecture on statistical methods for remote sensing. The next lecture from Colin will be on what are called semi-analytical methods for remote sensing. And in your lab today will be with Colin on using her simple semi-analytical technique to retrieve things from spectra. And then if time permits, at the end of the class, we'll have still another family of ways to retrieve things that are called spectrum matching techniques. Okay, so you kind of recall this plot where it's sort of the outline of the remote sensing or inverse radiative transfer problem, where you have incomplete light measurements. Maybe all you have is remote sensing reflectance spectra. That's what's coming back from your satellite, and you don't have all these other radiance measurements and irradiances and all of that. You just have this. So you may not know, this may or may not be a really good spectrum because you have imperfect atmospheric corrections. Maybe there's things like the waves on the sea surface, you don't know what those were. Well, hopefully they don't have a big effect, but at least you don't know what they were. And then you're going to have a relatively simple mathematics model that will relate what you do know, like the RRS spectrum, to what you would like to know, like IOP is chlorophyll, bottom depth, and so forth. And you may have to add some constraints in order to get a solution that actually makes physical sense. So we'll see how that works in a minute. But anyway, you go through this and you get an estimate of what you want. So IOP is chlorophyll, whatever. And I'll emphasize it's always an estimate, because in remote sensing, you're never 100% sure you actually got a good value. And so this estimate has the effects of all these errors in atmospheric correction, radiometric calibration at the satellite, on and on. Okay, so these statistical methods, they're one family of the math model that you're going to feed your data into and get out the answer. And essentially, they're just correlational models where you've got some data set of your inputs like RRS, and you've got some outputs like chlorophyll or absorption at some wavelength. And you basically just have a big fancy curve fit that, think of it as just a line fit through, some X and Y points where X is the chlorophyll and Y is your RRS spectrum. And you just come up with a best fit model. And you don't necessarily have any idea about the physics of why you got what you did. So for example, you may find that the chlorophyll concentration is correlated with the wavelength at 440 divided by the wavelength at 555. And there may be a very good correlation there that's very useful, but you don't have to understand why do those wavelengths tell me something about chlorophyll. If you do understand, good, but you don't have to know that, and you don't necessarily know it from these statistical models. So basically, we're going to have some generic sort of model in the mathematics, and it's going to have proportionality constants or weighting functions or fitting coefficients, whatever you want to call them. And we're going to determine those parameter values so that your model will fit the data as well as possible. And so that's where the statistics part comes in. and you just have best fit model for some data set that you've worked with. And so they're called statistical or empirical models. And then after you've taken some data that you have where you know the inputs and the outputs, force your model to fit it, determine these coefficients. Now you take the model with the coefficients and feed in new data and hope that you'll get out answers that look halfway correct. Well, we're going to look at two different types of statistical models, band ratio algorithms and neural networks. And band ratio algorithms are how this whole business got started way back in the 70s. So you've seen this, Ken showed it to you before. Howard Gordon and various other people went out, they measured, in this case, it's water leaving radiance for different chlorophyll concentrations. And then they simply looked at this and said, well, look, as we go from low chlorophyll, it's very high here at 443, and low chlorophyll, high spectrum, high chlorophyll, low spectrum at 443, but there's not much change in here. There's kind of a little hinge point. So they simply look at this and say, well, wait, if we took the ratio of, say, 443 to 520 or 550, that should be correlated with chlorophyll concentration. And they started out with a grand total of 33 data points from curves like this where they had measured the chlorophyll, and then this is ratio of band 1 to band 3, which is 443 to 550. They plotted the points, the log of the band ratio versus the log of the chlorophyll, and they got this nice little relationship here that was well fit by a straight line on a log-log scale. And with 33 data points, they started the whole business of ocean color remote sensing. Pretty amazing. Okay, so then you take these data points, you have a little model where you say, well, look, the log of chlorophyll is kind of coefficient plus another coefficient times the log of the band ratio. Least squares best fit, you'll get the values of C1 and C2, and then you can feed in a new ratio and come up and get what the chlorophyll was. About as simple as you can get. Okay, so they did that. They persuaded NASA to launch this coastal zone color scanner. And the idea is it would be useful for mapping things in the coastal zone. It actually worked much better in the open ocean than it did in the coast, but that's part of what they learned. So it should have been called the open ocean color scanner. Anyway, it had 4 visible bands, which you see here. And then there were 2 more out here in the infrared, near infrared, that they used for atmospheric correction, like we talked about on Friday. Okay, this thing over this period of late 70s and early 80s collected 66,000 images like this and totally revolutionized oceanography. And previous to this, you know, a lot of people kind of thought, well, you know, the ocean's just kind of homogeneous. It's just sort of, yeah, if you go from New York to Florida, there's a difference. But if you run around, you know, in the Gulf of Maine, it's all just looks like the Gulf of Maine. Well, obviously, there's enormous differences. There's all kinds of fine structure here and all kinds of little eddies and stuff. And people just didn't recognize how inhomogeneous the ocean is. And there's a fellow, Pete Umars, that he says, his little quote is, the ocean is inhomogeneous at all scales. You know, from 1 millimeter to the next, you're on top of a zooplankton or you're not, or from 1,000 kilometers to the next 1,000, you know, it's something very, very different. Okay, now as band ratio algorithms, that little one I showed you is what started it all, but it worked pretty well. And then of course, people start cooking up fancier band ratio algorithms. So here's the one, for example, the CWIF's OC4 version 4 to retrieve chlorophyll. So what that says is, look at the ratio of 443 to 555, 490 to 555, and 510 to 555. Look at those 3 ratios, take the biggest one, then take the log to the base 10 of that biggest ratio, and then plug that into this little formula, and you get back the chlorophyll. Now, what that is here's a sprinkling of the 3 band ratios, whatever they happen to be here, and the chlorophyll here. And you notice this is not exactly a straight line. That first plot I showed you with the 33 points, well, you know, it was a little area here and it looked like kind of a straight line. But you get more and more data and you realize, oh, you know, I could fit it better with some kind of little curve here, not just a straight line. So you start adding some terms, you have more data, you can determine more coefficients and on down the road. Here's the MODIS algorithm for KD 490. They look at 488 to 555, get that ratio, plug it into a little formula here, ratio to the minus 0.15 power, and you get back KD at 490 with some error bars. Here's MODIS's algorithm for absorption by CDOM at 440 and absorption by phytoplankton at 675. There they look at these 3 ratios again, are 3 ratios. So in this case, it's 412 to 551, 443 to 551, and 488 to 551. And then the CDOM absorption involves the ratios of the first band here, 1 to 5 and 2 to 5, and the phytoplankton ratio involves the 2 to 5 and 3 to 5 band ratios. And those are just the numbers of the MODIS bands. And you go through the literature, you can find these kind of algorithms by the dozen. And each one of them is basically like this one. That's just a best fit to whatever data they had. So keep in mind, with any kind of statistical model, your final model depends on what data set you had. Was it global? Was it for the Gulf of Maine? Was it for the Gulf of Maine in winter? And then, you know, you might get a great model for the Gulf of Maine in winter, and it won't be very good for the Gulf of Maine in summer. So whenever you use one of these models and you want to apply it to your own data, you need to go back and read the papers and find out what data set did they use to tune the model. And is that probably like my water body, or is it probably different? Okay, if you want to amuse yourself on a rainy Saturday sometime, here's a paper by Derek and Stramsky where they went through and they looked at all of these different band ratio algorithms that people have cooked up. So here's CZCS's for chlorophyll. You just saw that. There's a MODIS for chlorophyll. If it's case 1 water, there's a MODIS for chlorophyll. If it's case 2, another one, you know, MODIS is phytoplankton at 675. There's the CWIF's chlorophyll algorithm. What'll really be scary is pick the new case 1 model in hydrolite, do a run with chlorophyll of 2.3, you get an RRS spectrum, you know, look at the ratios of that hydrolite RRS at these different bands, plug all that in and retrieve the chlorophylls from these 3 different algorithms. And you put 2.3 into hydrolite, and you're going to get 1.5, 2.2, 4.1, whatever, depending on which algorithm you used. It's pretty scary because in principle, they should all give you the same chlorophyll, but they won't, I guarantee it. Okay, one of the virtues of band ratio algorithms is that you're throwing out the magnitude information when you take the ratio of the 2 bands. Now, here's a spectra, 2 spectra here. It's the same, this was actually an airborne measurement, but it's the same at sensor radiance, but atmospherically corrected by 2 different techniques, one called empirical line fit and one called TOFCA. We'll talk about those next week. But basically, one of them says, here's the spectrum after atmospheric correction. The other one says, here it is. They're different. They're like, whatever, 30% difference or so, or maybe even a factor of 2 down here. Well, when you take a band ratio algorithm, let's say you're looking at, you know, here we are, 440, and you're looking at the ratio of 440 to 555. The ratio of this number divided by this number may not be much different than this number divided by that number. So one of the virtues of the band ratio algorithms back in the days of CZCS, the atmospheric correction was really crude. it was actually basically pencil and paper almost, that the retrievals were not too sensitive to errors in the magnitude of the spectrum. So that was good. What's bad about it is you've thrown out some magnitude information. Okay, here's a bunch of spectra here. These all have the same chlorophyll value, which was less than, it was something like 0.018 milligrams per cubic meter. And you have entirely different spectra, but the ratio of this wavelength, the dotted line, to this one is the same for each one of those spectra. It's about 1.71 plus or minus 0.01. So in one case, you look at those spectra and say, well, yeah, those are obviously totally different, but they all look the same from a band ratio. So that means you end up getting non-uniqueness problems with band ratio algorithms. Now, this was actually a bunch of different water depths in the Bahamas, so the water was really clear. And the reason these are all different is they had different bottom depths and different bottom types, although the water column was the same. And so if you try to use a band ratio on this, you're just going to get something totally wrong in this case because it was contaminated by bottom reflectance. Okay, so a better example of that. So Heidi Dearson, who we would love to have down here for a guest lecture, but we're not allowed to this time. She was on this field experiment where those previous measurements were made. And she went around and made a bunch of measurements of remote sensing reflectance spectra versus depth in the Bahamas water. And then she plots that up, ratio of 555 to 670 versus depth, and gets a very nice curve here, fits a little function to it that works quite well for giving you back the depth of the water where you put in that ratio. Okay, so here comes Kurt Mobley. Oh, he finds a great formula in the book. Let's apply it to my data. Still in the Bahamas. Here's a sandbar. This is an island, the little red spot there, that's an island. This is a sandbar. This is a seagrass bottom out here. And the shallowest part in the sandbar is this deep because I went snorkeling there and I could just stand on the bottom and just get my nose above the water. And the tides down there are like this high, so not too much. So I take the Dearson algorithm, I apply it to each spectral in this, or each pixel in this image, and I do a perfectly good retrieval over the sandbar. So it says at the shallowest part here, it's like 2 to 4 meters deep. A little bit deeper area here, it's 4 to 6 meters deep. Perfectly correct. Out here, it says it's 0 to 2 meters deep. Totally wrong retrieval. So here's the real bottom bathymetry here. In some spots out here, it's as much as 10 meters deep. But the retrievals in here should all be saying something like 6 to 8 and then 8 to 10, and they're not. So what went wrong? Well, dig out hydrolite, do some simulations. I have some AC9 and other IOP data for this area, and then there's sand bottoms and grass bottoms and coral bottoms and so forth. So I did 4 hydrolite runs here. I took one set of IOPs, I put it as a sand bottom or a grass bottom. I took the other set of IOPs and I put in either a sand bottom or a grass bottom, and then I plotted Dearson's equation as the black line here. So you'll notice if we go back here, so I just showed right here a ratio of 25. So there's 10, 20, 30. So for this ratio of 25, the Dearson model says the depth is like, 4.7 meters or so. Well, I do the simulations here. So let's take the IOP set 1 sand bottom, and that's this red curve that's coming along here. And I come up on the ratio value of 25. I come up to the red curve and it says it's about 4 meters deep. Well, great. That's exactly what we're looking at here, sand bottom, and I'm getting a good retrieval. But if I take that same set of IOPs, and I put the grass bottom on it. So that's the area that was dark in the image and where the retrieval failed. Look what happens. So the ratio, this kind of yellowish color, comes up and kind of curves back. And so for a ratio of 25, I could either have a 4-meter depth bottom or I could have a 9-meter depth bottom. So it's a non-unique retrieval. And the reason that I got these shallow waters here was that it's coming up and saying, okay, here's the value on my curve. Ratio is 25, gives me back 2 meters or whatever it happens to be. Whereas the real answer should have been 8 or 10 meters. So there's a nice example of a non-uniqueness problem with the band ratio. And of course, the reason it failed for me is I took her model determined from her measurements, which were often these shallow areas, and it might have been a different stage of the tidal cycle or whatever. And I applied her model to my imagery, and I was pushing the model beyond where it should have been used. So nice example of non-uniqueness. Okay, when I actually plot these spectra now, so here's the IOP set for the bottom at 4 meters in red and the bottom at 9 meters in blue. You look at those two spectra and say, well, those aren't anything alike, but both of those have a band ratio of 550 to 670 of 25. So for the band ratio, the red spectrum and the blue one look the same, even though they're clearly different spectra. And then the green curve is the bottom reflectance that I used in hydrolite generating those two spectra. Okay, so the bottom line is if you have some data and you can kind of look at it and say, oh, you know, some kind of curve kind of like this should fit my data, or maybe it's a straight line, or maybe a second order curve or third order, whatever. You have some idea of what kind of math you should use to make up a little curve that would fit through your blob of data, and then you can use that model and determine the coefficients. But now, what if you don't have any idea what the mathematical form of your model should look like? What do you do then? Well, one thing you can do is neural networks. Now, about 12 years or so ago, when we were getting ready for this class, and I had been given this band ratio lecture, and the manual says, well, Kurt, you ought to also add a lecture on neural networks. And I said, well, like, who cares? Nobody uses neural networks in oceanography. There was like 2 papers had been published, and it just wasn't on anybody's radar except Emmanuel. But he was signing my paycheck, so okay, fine. So I go read up on neural networks and give a lecture on neural networks. Well, it turns out now, of course, the entire world is being run by neural networks. So Emmanuel saw it coming and I didn't. But anyway, here's the idea. Neural networks, and we'll see what this multiprocessor thing means in just a second here. And they're based sort of conceptually on how animal brains work, where you have all these neurons and they're all interconnected in some way or another. And some way that brain decides when I see an animal, it's a dog or a cat. So the idea here is you do very simple processing steps. So each little multiprocessor thing here, or each nerve cell in your brain, is doing something very simple. But all those processors or all your brain cells are all extremely connected with all the others. And you have simple inputs and outputs, so just real numbers. And you have, the important thing is adaptive interaction between the elements. So your brain can learn new things. You don't have a model that's just like fixed once and you just keep using it. So we'll see how this works in a minute. These are useful if you don't know the mathematical form of the model that links your inputs and your outputs. And you need to have lots of examples of the behavior that you want. So in other words, you need to have a lot of RRS spectra and a lot of the corresponding chlorophyll values. And you're going to use those to train the model to recognize chlorophyll from the input RRS spectrum, or to recognize what a dog looks like when you're given a whole bunch of different animals to look at. So we're going to determine the model structure from the data set that you have to train the model. So here's the idea in a brain, a real brain. You have a little simple processor here, the cell body, and it receives some inputs from these neighboring cells. And then all it has to do is decide, based on this input, do I fire and send a signal downstream to my neighboring cells, or do I not fire? That's all it amounts to. So it looks at these inputs and says, oh, wow, these are big enough or whatever. I'm going to send an electrical pulse downstream, and then all my neighboring cells will receive that, and then they can decide what they want to do with it. So here's a simple little neural network. You always have what's called an input layer. That's your input. So that might be remote sensing reflectance at 443 and RRS at 555, let's say. And then there's going to be a set of weights, which says in the processor, take the first input, multiply it by the first weight, take the second input, multiply it by the second weight, add those together, maybe add in some bias, a constant bias, and then if that sum is less than some threshold value, output a 0. If the sum is greater than or equal to the threshold value, output a 1. So that's all the neural network does. And then you get some output here that's either, in this case, 1 or 0, but in our case might be something like the chlorophyll concentration. So it's doing two things. It's doing a weighted sum of the inputs, and then it's comparing that sum with a bias perhaps to some threshold value in determining an output. So conceptually, very simple. The catch is maybe I'm feeding in RRS values here and I'm getting out chlorophylls, but how do I know what weights to use? Okay, so just some examples of neural networks in the real world now. When you use Google Translate or you tell Siri to, turn on the lights or whatever, that's all underneath some neural network that's recognizing your input and playing around with it and giving you the output a tenth of a second later. And I found that Google Translate, you put in some English, it comes back, does a pretty good job, even in things as different as English and Chinese. So it works well enough. I would have said that slightly differently, but this is a perfectly good translation of that as well. So it's not great literature, but it works. Here's, you ask Google, send me some cat photos. Well, it wasn't like some guy sat there getting paid $150,000 at Google headquarters to pull out cat photos and then send you a bunch of them. It's a neural network that has learned to look at lots and lots of photos from all kinds of animals and trees and people and houses and everything else. And it can some way spot, these are the cat photos. And now and then, it'll send you maybe a picture of an opossum or a skunk or something, but they do pretty darn well. And this goes back, this article was from 2012 when Google was putting no telling how many billions of dollars of research into neural networks and machine learning and so forth, and giving you things like that Google Translate program as output. And they never ever, some way built in what a cat photograph looks like. They trained the neural network from looking at lots of photos and then letting it learn what a cat looks like. So this is now just ubiquitous around the world, where you have, here's a street in, I think it was New Orleans. There's a camera, the camera's watching you walk down the street, and it's pegging you as an okay person or a wanted criminal or whatever. It's doing facial recognition and going back and looking through some database and saying, go arrest this guy, or no, this person's okay. Here's one inside a convenience store where it's simply spotting, this is a person, this is a person, this is a person, you know, and keeping tabs on people. And it's kind of interesting, you know, you read a lot of articles in the newspaper about how, oh, you know, you walk down the street in Beijing, you're being watched and like, you know, observed by all these cameras and all these neural networks, and they're pegging you as a terrorist or, you know, a tourist, whichever one it happens to be. So what do you think is the most heavily surveilled city in the world? Is it Beijing? Is it Moscow? Is it Pyongyang in North Korea? London, England. The most, you cannot like snatch a purse in London, England, anywhere in town without the police seeing you from like 7 different cameras, and you will be nabbed before you get a block. Now that's very good. It makes London a very safe place to walk around at night. But if you're the civil libertarian kind of guy that doesn't want the government tracking me, don't go to London. Okay, you now see hundreds of papers, artificial neural networks and medical diagnosis. Here's one on estimation of oceanic constituents based on the MERIS instrument. MERIS's retrieval thing is not some band ratio, it's not whatever, it's a big neural network. And I was talking to this fellow, Roland Dörfer, German guy who's a big name over there, involved in the MERIS things, and he's telling me, this is like 15 years ago, how MERIS is using neural networks to do their retrievals. And I said, well, that's great, but I mean, where'd you get all the data to train the network? He says, oh, we ran Hydrolite. And I just thought, oh my God, you know, if there's a mistake in Hydrolite and it's putting out bad spectra, I have blown away the entire MERIS project, you know? So that's what causes me to lose sleep. But he notes here in this article, the advantage of neural networks is not only that they achieve higher retrieval accuracy than more traditional techniques such as band ratio algorithms, but they also allow the inclusion of usually superfluous or unused information such as geometric parameters and atmospheric visibility. So incredibly powerful. On those band ratio algorithms, you didn't see anything in there about atmospheric visibility that would say, maybe don't trust this value too much because it was a hazy day. Emmanuel?
I mean, I think this is extremely valuable. And the reason that this other information is extremely valuable is if we use only optical information from now, it's like we know nothing about the ocean. It's as if we're on Mars and there's, you know, and we're looking at spectra and then we're inverting into whatever. But there's, you know, we know quite a lot of things about ocean ecology, both in terms of what was there yesterday versus what's today. Currently, we don't use information about what we measured yesterday. We don't even use information about the next pixel next to us. It's each pixel as its own, as if we completely don't know anything about the ocean. And that could be done much, much better. We don't use temperature in most of the ocean. Temperature is a huge constraint on ocean ecology. That long depth of water, all those are not used in the products you download from the website. So there's a huge room for new generation to just blow us away with much better products by using the power computers have to include much more information that's relevant.
Okay, and of course, like in the medical field, it bothers doctors, but a lot of times now, when you use a neural network, it often hones in on some weird little something that the doctor may not be thinking of, because it's completely objective and you've trained this neural network with 1 million different diagnoses and 1,000 different medical problems. And so I go in and I start telling the doctor like, oh, well, you know, I hurt here. And he says, well, yeah, you're 74, you're fat, you're lazy, et cetera. It's probably a heart problem. Makes sense. But actually it might not have been a heart problem. It might have been some like weird genetic thing that he wasn't thinking of or she wasn't thinking of, but the neural network in its training set says, oh, you know, these particular things, it might actually be this other thing. And so they often do as least as good a job and sometimes better than your average real doctor. Okay, So we have to train the neural network though before we can use it. And the very essence of neural networks is that they can learn from your data sets. And so the training simply means how do you come up with a set of weights that takes the inputs like R or S spectra and gives you the outputs like chlorophyll. So there's several ways you can do that. You can think of this big neural network is like an enormous equation with a bunch of unknown parameters, and you do basically a nonlinear least squares fit that says, give me a set of parameters that will do the best fit to my inputs and giving me the outputs. So it's just like a least squares problem. It's just enormously more complicated. Another way that we'll play with is called back propagation of errors. And so we're going to see how that works coming up right now. So I have a little pamphlet here somewhere. Yeah. Okay. So here's a neural network. It's got two inputs, X1 and X2. It's going to have two weights, W1 and W2. It's going to take those, sum them together. There might be a bias it'll add, and in this case, it says if the sum is less than 0.5, output a 0. If the sum is greater than or equal to 0.5, output a 1. So there's our neural network. And then we have our training data. So we have 4 sets of values to train on. So we have data set numbers 1, 2, 3, and 4. Here's the input X1 and X2, and here's the output that we want. So it says if both inputs are 0, I want to see an output of 0. If either input is 1 or they're both 1, I want to see an output of 1. So if I can some way come up with a set of weights that gives me those results, then I've trained the neural network. Okay, so the way this back propagation of errors works is we're going to take the inputs. We start out with an initial guess for the weights, and you can guess anything you want as long as the weight's not 0. So you start out, in this case, let's say 0.1 and 0.3 are the two weights. We're going to take the X1 value times the weight, which is 0.1, and that gives 0, and X2 is 0 times the weight is 0. Well, the sum there is 0, the threshold is 0.5, the sum is less than 0.5, so it outputs a 0. And that's the right answer. That's what we wanted to get. So there's no error. Okay, we don't do anything. Now we're going to go back to the next training set here, and we'll see how that works. So let me just click the button here. Let's see, I go to, hang on. Whoops, this thing's up here. So I want to, oh, there we are, next iteration. Okay, so we're going to go to the second value where the inputs are 0 and 1. So it says 0 times the first rate is 0, 1 times the corresponding weight is 0.3, the sum is 0.3. It's less than 0.5, so output is 0, but the right answer was 1. So then you have a thing called the learning rate. So when you get the wrong answer, you take the error, in this case 1, times the learning rate, 0.2, and you add that value into all the weights where the input was non-zero. So you say, okay, I had an error of size 1, learning rate's 0.2, so I'm going to add a 0.2 to the 0.3. That gives me a new weight of 0.5. This one had a 0 input, so don't mess with it. Okay, so now I have a new set of weights. They started out at 0.1 and 0.3, now they're 0.1 and 0.5. Now let's look at the third value of the training data. Okay, now the inputs are 1 and 0. So we take 1 times 0.1 is 0.1, and 0 times 0.5 is 0. Sum is less than 0.5, output is 0. Oh, I wanted to get 1 for the answer, so I have an error of size 1 times the training rate is 0.2, added into the weights where the input was non-zero. So 0.1 plus 0.2 gives me 0.3. This had a 0 input, don't mess with it. So now I have a new set of weights, 0.3 and 0.5. Okay, now go to the next value in the training set. So, whoops, hang on, I messed up now. I don't think I can back up here. Let's just start over. So I go 1. Okay, so now I go to the fourth thing where both of the inputs are 1. So, and there's my weights. 1 times 0.3, 1 times 0.3 is 0.6. That's greater than a half, output of 1. That's what I wanted. The answer should have been 1. I got 1. There's no error, so don't do anything. Now go back and work through the training set again. So we go back to the input set number 1 is 0 and 0. We get zeros, out comes a 0. We wanted a 0. Everything's fine. Do the next iteration. We go back and now try the second data set again. Inputs 0 and 1. We get a 0.3. Well, that's less than 0.5, so it outputs a 0. We had an error. Go back and add 0.2 to this, and we get 0.3 plus 0.02 is 0.05. New set of weights. Try the next one. So there's the third one. So now we're getting 0.3s. We output a 0. Oh, we should have had a 1. Take 1 times 0.2, add it here. Now we have a set of weights of 0.5 and 0.5. Go now, look at the fourth one again. Okay, we input 1 and 1 times point times our weights. That's 1. It outputs a 1. That's what we wanted. Error 0. Don't do anything. Now go back and go through the training set again. So now we're inputting zeros. We're getting zeros. Fine. We check the second one. Well, we still aren't quite there. It inputs the 0.5. It needs to be greater than the 0.5, so it outputted a 0. So take 0.2 plus 0.5 gives 0.7. Try the next one. Okay, that one gives the right answer. Whoops, let's see here. Hang on.
Yeah, that's right.
Yeah, so now we're getting with our 0.7s, we get the right answer. Go through the training set again. Back to zeros, we're getting out of 0, we got the right answer. We do the next one, we got a 1, okay, that's good. We do the next one, we got a 1 again, no error. The last one gives us no error. So now I've got a set of weights here, which in this case is 0.7 and 0.7, that completely reproduces my training set. So I can keep going through the training set now as long as I want, and it will never change those weights. So that's what the training of the set looks like. And normally you're going to have here, you know, RRS at 5 different wavelengths, and you'll have 1,000 values, and you'll have, you know, 3 outputs, which are chlorophyll, absorption, 440 or whatever. And you're just going to let the computer go through this training set thousands of times and finally come up with a set of weights that does a pretty good job of fitting your data. Now, there's a little catch, of course. You should have been so lucky that it would be that easy. Okay, so what you notice is that the neural network was able to use this training data to come up with a set of weights that produce the desired output, and that's the training. And the knowledge or the memory of the neural networks is contained in the weights. Now, if you have a real world problem, you're never going to get perfect predictions of chlorophyll. What you do is, whoops, wrong direction. Okay, that's, hang on. Maybe Yeah, okay, I see where I'm going. Okay, so here's a more realistic real-world example from a paper including Wayne Slade, one of Emmanuel's grad students, in which they started out with 1,100 sets of RRS spectra and the corresponding chlorophyll values that they pulled out of these various archives. And this is why you need to upload your data to the archive when you get funded by NASA, is so people can use it for other things. So they constructed a neural network that had 5 inputs, which was RRS at 5 different wavelengths, had 2 hidden layers, and we'll see those in a second, and 1 output, which is chlorophyll. Then what they did was they took their 1,100 sets of data and they said, let's use half of them for training, a fourth of them for what's called validation, and then a fourth of them to actually test how well the neural network did. And you should just like randomly pick these spectra out of your data set. There's nothing magical about using half, a fourth, and fourth. You could have used, a third, and a third if you wanted to. Okay, and then they compared the neural network's predictions of chlorophyll with those from this CZCS OC4 version 4 band ratio algorithm. So here's what their neural network looked like. Their input layer is RRS at 5 different wavelengths. They used 2 hidden layers that had 6 processors in each layer here. Why 2 of 6? Why not use like 1 with 12? Or why not use 4 layers with 3? Answer is it's kind of a black art. You want to have some layout here where all these neurons can talk to each other, and like everything in this layer receives input from each of the inputs. And then this layer, each one of these elements talks to each one of these, and then finally these guys output a value, which is going to be the chlorophyll concentration. So there's no way that says, I have this problem, this is how I should design my neural network, and I should have 2 layers of 6 instead of, 3 layers of 4 or something like that. And a lot of computer science people and mathematicians spend a lot of time trying to figure out, what's the best design for a neural network. Yeah.
So it does make a difference if you have 1 layer of 12
Right.
versus 2 of 6, because by moving it from 1 of 12 to 2 of 6, the implication is that the layers are impacting each other. They're in a different way than they are in the same layer. So by adding those and some of the knowledge of how each other, right? I mean, so there's an art to it, but it's not just mathematical art. There's an underlying understanding of the processes that impact remote sensing and reality.
Yeah, the bottom line is you want to have these guys all talking to each other. You want to have all of these are getting input from all of these, which if there was 1 layer of 12, they would all get inputs here and they would all give an output, but they wouldn't be talking to each other. And so you really have here 5 things in the input layer, 6 here, that gives you 30 weights here. There's 6 times 6 is 36 weights. And then there's 6 more weights here where this guy decides whether or not to fire. And then those finally all get turned into the output. So yeah, Ben.
I had a question about 2 slides ago. In a more complicated situation with balance having enough neurons to capture the science, but not so many that the neurons are the noise of the training data.
Okay, glad you brought that up. As Nixon said once, I'm glad you asked me that question. So here's what's going on with that statement. When you have your training set and you just keep cycling through it time after time after time, it keeps tweaking the weights a little bit. Well, at the start, you're really fitting the biology or the physics, but your training set has noise in it and errors and whatever. So as you keep going through the training set, you'll keep getting a better and better and better fit to your data. But somewhere along the way, you're now starting to fit the noise in your training set, and you don't want to do that. So the reason they used half of their values for the training, and they took a fourth for what they call the validation set. So you go through cycles here of working your way through the training set, and you keep getting a better and better fit. So this is mean square error between the outputs and the correct output. And then you back up and you say, now let's take this fourth of the values that we used for validation and apply the neural network to those. And that's independent data. So those values start getting better as well. But then somewhere along the line, maybe after 1,000 passes through the data, maybe after 1,000,000 passes, the training set will keep getting better and the validation set will start getting worse again. And the reason is because up to this point, you're tweaking your weights and they're describing the physics or the biology. And then when you start fitting your training set really to the noise, now the noise is different in the validation set, so it starts getting a little bit worse. So what you do is you run the neural network, and then when your validation set starts getting worse, you back up and say, this is the point where I want to stop and use these weights to now go do science. And then you take got your training set, your validation set, and then you use that last fourth of the data to now say, take these weights right here, apply it to this last bit of independent data and see how well I did. Okay, so here's their results. Here's the measured chlorophyll, here's the CWIF's retrieved chlorophyll, and here's the spread of points. When they use the neural network on the same image, here's the chlorophyll, here's the neural network retrieved values. You just look at that and say, well, you know, that's a better fit than this is. So the neural network has done a better job of retrieving chlorophyll from the CWIF's image than the CWIF's algorithm did. And in fact, here's on the testing data, the CWIF's algorithm had a root mean square error of about 0.5 milligrams chlorophyll per cubic meter, whereas the neural network was only 0.2, considerably better job. Okay, so here's the actual image. Here's the CWIF's chlorophyll, and this is, let's see, generated by applying a neural network. So there's their retrieval. And then here's the difference of the neural network minus the CWIF's values. So you see here, there's areas where the chlorophylls are down, you know, less than 0.5. And the difference here is this kind of greenish color. They're pretty much in agreement. On the other hand, there's spots up along the coast here where the difference in the CWIFs and the neural network, that's the dark blue, that's like 1, 1.5, maybe even now and then 2. So here the chlorophyll values were like 0.5, or no, sorry, if we're right in here, they're like kind of that greenish color, 1.5 or 2, but the errors were sort of 1 or 1.5. So percentage-wise, very big errors along the coast here. Out in the open part of the area, the errors were pretty small. They were in good agreement. So the reason the neural network did so well is it was doing a better job of these near coastal areas here where it's probably a lot of CDOM or mineral particles or what in the water. The case 2 water, the CWIF's algorithm was designed for case 1 waters. So it did a poor job right next to the coast, but the neural network says, man, I don't care anything about case 1 or case 2. You just gave me RRSs and what the chlorophyll was, and I tweaked the weights, and here we go. So it did a much better job in the areas where you may be most interested, which is right next to the coast. And yeah, out here, the off-the-shelf algorithm worked just fine. Errors are pretty much 0. there's a little too high there in that patch right there, but okay, fine. So that's the story. So there's a lot of websites, of course, that tell you about how neural networks work. There's one I ran across, it's called 3blue1brown.com. And they have all these tutorials on just almost anything imaginable in mathematics. And there's a whole big section there on neural networks. And so I stole one of their examples, which is, let's say you have a little touchpad and you want to train it to recognize numerals 0 through 9 as you write them there with your finger. Well, you can think of, here's a little touchpad, it's got 28 by 28 pixels, and you're gonna use your finger and just write a 3 or a 4 or something on that and have it recognize it. Well, how does that work? Okay, just think of the 2D grid here, just unwrap this and make a long set of inputs. In this case, 28 by 28 is 784 inputs. And the input is either going to be, it's a 0, he didn't touch it, or it's a 1, he did touch it with his finger. So you have all these inputs. Then they have some hidden layers here with a gazillion connections between 784 inputs and whatever that is, like 15 inputs or so. And then they train this on lots and lots of different shapes of numerals and so forth. And finally, this thing says, if there's a certain pattern of ones in here, it looks like what I'll call a 3. And that's all it amounts to. And it works amazingly well. Some of my Chinese friends now on their iPhones and stuff, they have apps where you can just use your finger and put in the Chinese characters, and then it will identify it and come up and give you like the printed version of the character. So you can basically use your finger to put in the characters, and it will then generate an email, for example, that has the characters, and it works amazingly well. It just blows me away. So anyway, you can read up on that if you want. The take-home messages there are that these statistical methods, they work very well. They're still used. That's what Modis and Veers are using for all different outputs. They're only as good as the underlying data that you use to determine the coefficients, whether it was a neural network or whether it was just a simple equation. You always have to look at the data. And the statistical nature of the algorithms often obscures the underlying biology or the physics. Okay, they're operationally useful. Intellectually, there's kind of nothing new here. You know, we know, a dozen different band ratio algorithms to retrieve chlorophyll. And I have no interest in myself going and getting a new data set, fitting a new band ratio algorithm. Intellectually, it's a kind of solved problem. But then there are new techniques like neural networks and, all the buzzwords of machine learning, deep learning, all this kind of stuff. It's all basically a bunch of neural networks underneath. Okay, so one time I'm captain. Yeah, Emmanuel.
Just about confirmation bias, I was totally against this, 20 years ago. I thought this is a waste of our time. I thought we don't learn anything from them. It's just black boxes. I almost did not hire Wayne to be his master on this because I just could not believe a scientist would use these kinds of methods because you learn nothing. What do you learn from them? And I turned completely in the last, probably 10 years to think that this is the future. Because yes, you're right, we might not understand the physics, we might not, but the proof is in the pudding. And I believe this is much better than our limited brain, with our limited understanding can do. So I was one of those, it's a black box, what science is that? An engineer's okay, but science is not. And
Yeah.
The thing about it is that it removes your bias, right? So if you think about the classification algorithms for the ISP, and you look at it, and you might be really good at identifying one kind of diagram, so you are perfect. You have classified that one, you nailed it. But then, the other spherical, let's put an L. Okay, but It doesn't have that feeling of like, the diagrams are so beautiful. Over-classify them, don't worry about the diagrams, right? So it doesn't have that bias in it. And so it is a far superior way to classification.
Yeah.
And in fact, data we can approach.
We just
When
don't know,
we started
we
in the
don't know
field,
why, right? We don't know why it's classifying that dynamo. We don't know what it is about that dynamo that made the network say, that's it. And that's the frustration.
we can investigate, you can investigate the weights.
Investigate,
We can go
it's
back
not
to-
as like,
When we got in the field, people use Excel to do remote sensing. And obviously nowadays, you know, my students are in the supercomputers for many of their problems.
And one thing you cannot do is after you determine all your weights, you go back and look at the weight between input number 3 and hidden layer thing number 5 and this number 6 and all these weights, well, that must be what's going on. You really can't do that. You just have to take the weights as a big set of numbers that gives you a good answer. But you can't go back and look at a particular weight and in some way say, oh, that one's telling me how 440 wavelength gives me a good absorption and this other wavelength is how 555 is giving me a good backscatter. That's pushing it beyond the math. Okay, well, to finish off today, I'm poking around down in the bottom of the Grand Canyon and I find this rock. It's one of the most beautiful rocks I've ever seen. And up above me, a couple hundred feet, there's a big layer called the Moab Limestone. And it's kind of just generally gray limestone, not too attractive, but a piece of it had broken off, rolled down by the river. The water had polished this rock over the years. And this is about, let's say, a foot high. It's kind of like this big of a rock. And I noticed in it these red layers. Those are fossil algal mats. So this Moab limestone, it's early Cambrian, 500 to 525 million years old. And this is just your basic limestone laid down by layers of mud in a little shallow sea. And then here and here and here, you see these blobs of the red stuff. And living organisms take up isotopes of oxygen and nitrogen and phosphorus and all those things at slightly different ratios than you see in the environment. So they've actually looked at the isotopic ratios within the red layer versus out here and concluded that these were probably layers of scum on the bottom. They got buried, they got fossilized, and here they are today. So it's just an astounding, to me, fossil to see and think, well, 500 million years ago, this guy was like the smartest guy on earth. We hadn't had anything crawl out of the ocean yet and poke around on land. So it was really interesting. And, I would have given anything to have this rock on my desk at home. But first of all, it's illegal to take stuff out of national parks. And B, I was a Sierra Club leader of this trip. So it would look really bad to see in the newspaper, Sierra Club leader arrested for stealing rocks out of Grand Canyon. And third is it actually goes down below ground and I wasn't able to get it out. So this may be like the tip of a boulder the size of this house. But anyway, the reason I was down there was to lead a Sierra Club raft trip. And this is one of those like trip of a lifetime that you should do every year, but at least do it once. And I've done it, the raft trip, a couple of times, and you want to do the full thing. It will take you like 14 or 16 days. Don't do like halfway and hike out or hike in and do the second half, because the upper half and the lower half are much different geology, flora, fauna, etc. And you don't want to go through on a motorized boat where they got like 30 people and they run you through in 4 days. Get a paddle raft trip and really get down at river level and experience it. So here's a sequence of shots. I'm standing on a rock next to what's called Lava Falls, which is the largest runnable rapid in North America, class 5 rapid. Here's about half of the oceanography community here. And so there's these big waves. They're like, 10 or 12, 15 feet high. These rafts are loaded down with all your food and gear for 2 weeks. So the rafts don't ride up and over the waves. When you hit a big wave, you actually punch through it and you go under the wave and hopefully come out the other side. So here these guys are, there's 6 paddlers here and 1 guy in the back steering. They're getting ready to go into a big wave. So here's this enormous wave. They go down into this hole, hit the wave head on, basically completely disappear for a few seconds with like 10 feet of water over their heads. And then they pop out on the downhill side and you count heads. Well, actually, amazingly enough, there's still 7 people in the boat, which doesn't always happen. And you come out and you go through a series of these waves like this, and then you come to Dead Man's Rock. And you've been coming down here and you have to take this quick left turn. It drops down like another 10 or 15 feet in elevation. And then once you get down here, you're done, you're okay. And the interesting thing is in Dead Man's Rock, about 10 or 15 feet underwater here, there's a tunnel that has been worn by the water through the rock. And this rock is, like half the size of this room. And when you come out around the bottom side and look back, you actually see the daylight coming through the rock. And there's actually been at least 1 person I know of that fell out of the raft in the falls and was sucked through the rock and poked out the other side with minor cuts and scratches. It's like, talk about luck. But anyway, that's the Grand Canyon. You definitely want to do it. Do it in either April or May or September or October, because June, July, and August are way too hot to be in the Grand Canyon. It's like 120 degrees down there. So that's it. So it's 10:20 now, Colin. You want to do 10:30 start? Okay.
Don't forget we have testing today. So, yeah, if you don't finish by 11:30, you may not. Yeah. So we'll come start at 1. Today. We can get early. Thank you, Lucia. Very helpful. Appreciate it. Yeah.
Yeah. No, I wasn't in the phone. I was taking notes on those. So no photos.
Or I was just like,